Welcome to Elucidate. Today we are working through the first lesson of DNA, the code of life. We are going to look at where DNA lives in the cell, what a DNA nucleotide is made of, and how the molecule is put together. For each idea, I'll introduce it and explain it, and then we'll work the Scratchpad together, so you can see the completed step on screen. By the end, you'll be able to locate DNA, build a nucleotide, pair the bases, label exam diagrams, and explain why DNA is the carrier of the genetic code. Let's begin. By the end of this part, you'll be able to tell a nucleic acid from a nucleotide, and name the two nucleic acids. Here's the key idea. There are exactly two nucleic acids in this topic, DNA and RNA. Both are large molecules, built from smaller repeating units. That repeating unit is the nucleotide, one phosphate portion, one sugar portion, and one nitrogenous base. So the whole point is scale. A nucleotide is a single building block. A nucleic acid is the long chain built from many nucleotides joined together. Keep the sizes straight, and the multiple choice answers itself. DNA and RNA are the chains. A nucleotide is one link in the chain. In the Scratchpad, the first stage checks this exact distinction. The question asks, a nucleic acid is best described as, and then, a nucleotide is best described as. The answer, a nucleic acid is a large molecule made of repeating nucleotides. And a nucleotide is one phosphate, one sugar and one nitrogenous base. Both answers are now marked correct. By the end of this part, you'll be able to name every place in the cell where DNA is found, and write the exact structures the exam wants. Here's the key idea. Most DNA sits in the nucleus, where it makes up the genes on the chromosomes. That's the nuclear DNA. But DNA also lives outside the nucleus. Mitochondria carry their own DNA, and in plant cells, the chloroplasts do too. This is a classic mark loser. If you write that DNA occurs only in the nucleus, you lose the mark. The exam wants the exact structures, mitochondrion and chloroplast. One more thing, ribosomes and vacuoles carry no DNA at all. So when you tick the structures, tick nucleus, mitochondrion and chloroplast, and leave the rest. The Scratchpad asks two location questions. First, tick every cell structure that contains DNA. And then, name two places besides the nucleus where DNA is found in a plant cell. The answer, nucleus, mitochondrion and chloroplast contain DNA, ribosomes and vacuoles do not. And the written answer is, mitochondrion and chloroplast. Those are the exact words the memo wants. By the end of this part, you'll be able to name the three parts of a DNA nucleotide, and label them on a diagram. Here's the key idea. A DNA nucleotide has exactly three parts, a phosphate portion, a deoxyribose sugar portion, and one nitrogenous base. They always arrange the same way. Phosphate at the top. Deoxyribose sugar in the middle. One base attached below. And a base alone is not a nucleotide. That mix-up, calling the base the whole unit, shows up in multiple choice year after year. The base is just one of the three parts. Learn the three in that order, and the diagram labels take care of themselves. The Scratchpad builds a nucleotide from its three parts. The question asks you to choose the correct part for each of the three slots, then identify the parts on the diagram. The answer, slot 1 takes the phosphate portion. Slot 2 takes the deoxyribose sugar portion. Slot 3 takes one nitrogenous base. And the diagram labels run in the same order, phosphate, deoxyribose sugar, nitrogenous base. The stage is now marked correct. By the end of this part, you will be able to write the complementary strand for any sequence and name the bonds that join the two strands. Here's the key idea. DNA has 4 nitrogenous bases: adenine, thymine, cytosine and guanine, A, T, C and G. They pair by one rule only, A with T and G with C. A never pairs with G, T never pairs with C. You never guess the partner, the rule gives it to you. And the two strands are joined between the paired bases by weak hydrogen bonds. The word weak matters in the memo. The backbone is held together by covalent bonds, but the strands themselves join through the weak hydrogen bonds. One rule, one bond type, that's the whole stage. The Scratchpad gives you one strand and asks for the partners. Strand 1 is A, G, T, C, A. Choose the base that pairs with each position, then say which bonds join the two strands. The answer, the complementary strand is T, C, A, G, T. A pairs with T, G with C, T with A, C with G, A with T. And the strands are joined by weak hydrogen bonds. All 5 positions and the bond question are now correct. By the end of this part, you'll be able to read a stick diagram of DNA and label it exactly the way the memo does. Here's the key idea. In a stick diagram, each strand alternates sugar and phosphate along the outside, while the bases project inward between the strands. The exam labels test that pattern. The circles and hexagons on the outside are the phosphate and the deoxyribose sugar. The blocks between the strands are the nitrogenous bases. And the dashed lines joining the paired bases are the weak hydrogen bonds. Read the pattern before you answer. Sugar, phosphate, along each strand, bases paired in the middle. Once you can read that pattern, the labels write themselves. By the end of this part, you'll be able to give observable reasons, evidence you can literally see in the diagram. Here's the key idea. Observable means what the diagram shows you, not what you already know about DNA. Writing that DNA contains genes is knowledge, not evidence, it earns no mark. What can you see? The molecule is double-stranded, 2 strands are drawn. You can see base pairing, the bases are joined in pairs between the strands. And if the diagram shows the bases, you can name a visible base, like thymine, as evidence, because you can see it right there in the diagram. Evidence from the diagram. That's the whole skill. This is the real diagram from the 2025 Northern Cape September paper, question 1.4. The questions: identify nitrogenous base B, and molecule C and D and name the bond that joins A to B. Then give 2 observable reasons why the molecule is DNA. The memo answers: B is adenine. C is the deoxyribose sugar. D is the phosphate molecule. The bond joining A to B is a weak hydrogen bond. And the observable reasons: it is double-stranded, and it contains thymine instead of uracil. Every part of the question is now answered correctly. By the end of this part, you'll be able to match each structural feature of DNA to the job it does in the cell. Here's the key idea. The sequence of bases along the strand carries coded information for protein synthesis. The order of the bases is the code. Next, DNA makes up the genes on the chromosomes, and genes carry hereditary information from parents to offspring. That's heredity. And the double helix shape, with its 2 strands, makes the molecule stable and protected. The structure exists to protect the code it carries. Feature to function: sequence gives the code, genes give heredity, the double helix gives stability. By the end of this part, you'll be able to compare DNA and RNA across strands, sugar and bases. Here's the key idea. 3 comparisons decide the mark. First, the strands. DNA is double-stranded, RNA is single-stranded. Second, the sugar, DNA carries deoxyribose, RNA carries ribose. Third, the base, DNA uses thymine where RNA uses uracil. Thymine is DNA's answer to RNA's uracil. Set the table up as 3 rows, strands, sugar, base, and the comparison question becomes a straight fill-in. By the end of this part, you'll be able to attach the discovery history to the right names. Here's the key idea. Rosalind Franklin produced the X-ray evidence of DNA structure, the famous photo 51. Maurice Wilkins worked with the X-ray studies and contributed further evidence alongside her. And Watson and Crick used that evidence to propose the double helix model. Evidence first, model second. That's the order the memo expects. Franklin and Wilkins gathered the evidence. Watson and Crick built the model from it. Don't swap the names, the multiple choice item is built on exactly that swap. The Scratchpad consolidates all 3 ideas in one stage. Match each structural feature to its function. Complete the DNA versus RNA table. And pair each scientist with their contribution. The answers, the base sequence carries coded information for protein synthesis. Genes on chromosomes carry hereditary information. The double helix makes the molecule stable and protected. DNA is double-stranded, with deoxyribose sugar and thymine. And on discovery, Franklin produced the X-ray evidence, Wilkins worked with the X-ray studies, and Watson and Crick proposed the double helix model. The whole stage is correct. By the end of this part, you'll be able to answer describe questions the way the memo marks them. Here's the key idea. The command verb decides the answer. Name, or identify, means one word, the exact structure. Describe means state what you see about the structure. For a describe question on DNA, the mark-bearing statements are, a double helix, made of 2 strands of nucleotides, with bases paired and joined by hydrogen bonds. Write complete sentences that name the key structures. The marker looks for those propositions. Your exact wording can differ, but the structures must be named. By the end of this part, you'll be able to answer explain questions with a mechanism, cause and effect. Here's the key idea. Explain is harder than describe. You must link a structure to what it enables in the cell. 2 standard explanations carry this topic. The sequence of bases carries coded information for protein synthesis. And DNA makes up genes, which carry hereditary information. Build the answer in 2 moves. Name the structure. Then state what it enables. A single word will not earn the marker wants the mechanism. The Scratchpad gives you 2 real exam questions to answer in full sentences. Describe the structure of a DNA molecule. And explain why DNA is suited as a carrier of the genetic code. The answers, the structure is a double helix of 2 nucleotide strands, with bases paired and joined by hydrogen bonds. And the explanation, the sequence of bases carries coded information for protein synthesis, and DNA makes up the genes that carry hereditary information. Both answers are accepted, full marks. By the end of this part, you'll be able to attack a full DNA question in any orientation. Here's the key idea. Exam DNA questions follow one pattern: name the molecule, label the parts, give observable evidence, then state a function. Recognize DNA by its pattern, however it's drawn. 2 strands. Phosphate and sugar units along the outside. Bases paired between the strands. Whether the diagram is vertical, horizontal, or rotated, the same parts appear. Answer from what you see. The marks follow the visible evidence, exactly as they did in the labeling stage. The mastery check is an original exam pattern question on a rotated diagram. Name the molecule. Identify the 3 label parts. Give one observable reason from the diagram. And state one function of the molecule. The answers: the molecule is DNA. Part 1 is the phosphate portion. Part 2 is the deoxyribose sugar. Part 3 is the hydrogen bond. The observable reason: it is double-stranded. And the function: it carries coded information for protein synthesis. 6 out of 6 marks. You've worked through the whole Scratchpad. You can now locate DNA in the cell, build a nucleotide, pair the bases, label exam diagrams, relate structure to function, and write the explanations the way the memo marks them. Practice the Scratchpad at your own pace, run every question until you can answer without the hints. Structure first, explanation next. That's the whole exam route for DNA. Well done, and I'll see you in the next lesson.